so much for having me, and I am very excited to be here. And thank you everyone for making it for today. As Evgeny mentioned, I am Piotr Konieczny, and I am a postdoctoral fellow in Dr. Shruti Naik lab at Icahn School of Medicine at Mount Sinai. We just recently moved from NYU Grossman School of Medicine. And so today I will talk to you about a recently published story, which really center around this concept and the crosstalk between two different tissue compartments that really drive metabolic codependency, which is required for tissue pathology. But before that, before I will move to very specific experiments, I just want to give you a very broad perspective about from where this story came from. So as you know, every single day, our organs or tissues and cells are exposed to different environmental inputs like microbes, allergens, pollution, different types of the injures, in general, macro environment. And these environmental inputs really dictate our state of health and diseases. And very often they are experienced in our barrier tissue like the lungs, the gut, and the skin, which I will mostly focus today during my presentation. And this really environmental inputs are sensed by the immune cells, by our homeostatic immune cells that hang out in our barrier tissue. So the immune cells are very important sensors and mediators of these environmental inputs, and they engage with the crosstalk with non-immune cells, with other tissue cells present in the tissue. And this crosstalk is what really mediates tissue adaptation and maladaptation to these environmental inputs. So during my postdoctoral training, I was very interested and focused on understanding how homeostatic immune cells that are present in our skin engage with the crosstalk with skin epithelium, and how this crosstalk drive tissue adaptation, skin adaptation to damage. And we previously described tissue adaptation mechanism, which drive this crosstalk between immune cells and epithelial cells, which is required for optimal tissue repair. So we found that subset of homostatic lymphocytes, which we call gamma T, gamma delta T cells, they rapidly respond and proliferate during acute skin injury, and they express interleukin-17, which is directly sensed by skin damaged epithelium through interleukin-17 receptor. And what I want you to remember from this slide is that interleukin-17 is required and necessary to activate HIF-1 alpha transcription factor, so hypoxia-inducible factor, that really is mediate and is required to activate glycolysis metabolism and glycolysis gene expression, which really drive proper tissue repair and barrier restoration. So we really wonder whether the same mechanism and the same crosstalk which drive physiological optimal tissue repair can be co-opted in tissue maladaptation, and if this mechanism can drive damage repair pathology, like for example inflammatory skin diseases. So today I will discuss this concept. So as you know, tissue damage is a center to tissue pathology, and we have a very wide range of different clinical conditions where we observe this damage repair failure, like chronic wounds, cancer, which we very often refer as a non-heal wounds, or epithelial inflammatory diseases, for example, psoriasis, where we observe this overhealed phenotype where the cells actively proliferate. We have a massive immune infiltration. And one of the most common mechanisms that drive this damage repair failure pathology is this crosstalk between hyperreactive immune cells and our epithelial barrier. And this interaction causes severe damage and dysfunction of tissue epithelium. So we really wonder whether the same mechanism, the same signaling which we described in the context of physiological repair, which involved interleukin-17 and HIF-1 alpha signaling axis is co-opted in tissue pathology, and if this mechanism can drive inflammatory epithelial diseases. So first, we took a very unbiased approach. We leverage single cell data from Human Cell Atlas. We reanalyzed single cell data from skin epithelium from healthy patients, as well patient that with psoriasis or atopic dermatitis, so two most common skin inflammatory diseases. And we found that in skin inflammatory conditions, we see this expansion of a cluster, which we called a differentiated cluster 2. And when we look at the genes that are highly expressed, that define this cluster, and we perform pathway analysis, we found that this cluster significantly highly expressed genes that are involved in HIF-1 alpha signaling pathway, as well glycolysis metabolism. So this suggests, we were very excited to see these results, and this really suggests that maybe the same mechanism, interleukin-17 HIF-1 alpha, is involved in tissue pathology in inflammatory skin diseases. So with collaboration with two fantastic scientists in the lab, Dr. Ipsita Subudi and bioinformatician Alexander Prustyba, we really decided to focus on psoriasis as one of the most common skin inflammatory relapsing diseases that affects around 2 to 3% of population, which manifests by this red plaques, very thick red plaques. And the main reason why we decided to focus on psoriasis is that psoriasis is mainly driven driven by type 17 immune cells, which are hyperreactive, and they express interleukin-17 that cause this hyperactivation of skin epithelium and drive tissue pathology. So first, we wanted to see to validate our single cell analysis to look at HIF-1 alpha expression if HIF-1 alpha is present in psoriasis. So we leverage human skin biopsies, either from non-lesional skin or lesional skin. So non-lesional skin is the skin that is not affected by psoriasis, and lesional skin is active with active psoriatic plaque. And we found that indeed HIF-1 alpha is highly expressed in skin epithelium in the psoriatic sample compared to non-lesional skin. And this expression correlates with type 17 immune infiltration. So this really suggests that this crosstalk, which we observed in physiological repair, which involved type 17 cells and skin epithelium, maybe is essential to drive HIF-1 alpha expression in psoriasis. So to define if this axis, which involved interleukin-17 and HIF-1 alpha, is co-opted in psoriatic pathology, we took two different approaches. The first, we look on HIF-1 alpha expression as well interleukin-17 receptor expression between in a non-lesional skin and lesional skin, and we found that HIF-1 alpha is highly expressed in the cells that highly express interleukin-17 receptor. And the second approach, which we took, we leverage human punch biopsies, skin punch biopsies from patients that received anti-17 treatment, secukinumab. So we looked at HIF-1 alpha expression as well, interleukin-17 receptor expression before and after treatment. And we found again that HIF-1 alpha is highly expressed in skin epithelium in patients before treatment, as well interleukin-17 receptor. And after treatment, we observed significant decrease of HIF-1 alpha expression together with the expression of interleukin-17 RC receptor. So this really suggests that HIF-1 alpha is a downstream downstream of interleukin-17 signaling, and it really suggests that this axis is co-opted in tissue pathology, in psoriatic pathology. So next, we really wonder whether HIF-1 alpha is significant, important effector of interleukin-17 signaling in psoriasis pathology. So To validate, to define the function and contribution of HIF-1 alpha in psoriasis pathology, we perform a small clinical trial on the dish. This is my favorite experiment from whole paper, where we took skin punch biopsies from patients with psoriasis or patients with patients with healthy skin, and we culture this punch biopsies for 24 hours ex vivo on the dish in the medium, either with vehicle control, standard of care. So standard of care, this is corticosteroids that are very often used in the clinic to treat psoriasis, or with HIF-1 alpha inhibitor. And after 24 hours, we collect the samples, we isolate RNA, and we perform RNA sequencing. And here on this volcano plot, you can see the genes that are downregulated in samples that were cultured with HIF-1 alpha inhibitor, psoriatic samples that were cultured with HIF-1 alpha inhibitor versus control. So we observed this dramatic downregulation in HIF-1 alpha dependent genes, and we project these genes, so this signature, HIF-1 alpha signature, on a publicly available clinical trial where patients were treated with anti-IL-17 treatment, and we were very excited to see that HIF-1 alpha signature signaling distinguished anti-IL-17 responsiveness from placebo group, as well as stratify therapy responsive versus non-responsive responsive patients. So this results really proved that HIF-1 alpha is a downstream effector of interleukin-17 signaling, and that HIF-1 alpha transcriptional profile play a crucial role in psoriasis development. And this program, which we observed in physiological repair, can be co-opted in tissue pathology. And in this specific context, HIF-1 alpha act as an inflammatory factor, which is required for psoriasis development. So to define the mechanism by which HIF-1 alpha drive psoriasis development and psoriasis pathology, we turn on to our favorite mouse model, which we called imiquimod-induced inflammation models. This is model which is very well established in the field, where we treat mice, the back skin of the mice, with imiquimod for 6 days. Imiquimod is a Toll-like 7 receptor agonist, which induced psoriatic-like phenotype in the mice, in the skin of mice after 6 days. And here you have just example of this psoriatic plaque, where we see as well that HIF-1 alpha is highly expressed in skin epithelium. So to define the function and molecular function of HIF-1 alpha in psoriatic development, we generate HIF-1 alpha specific epithelial specific knockout mice, and we found that by blocking HIF-1 alpha specifically, inhibiting HIF-1 alpha specifically in skin epithelium, we almost completely broke psoriasis progression and psoriasis development, and keratinocytes skin epithelium hyperactivation, which we define by epidermal thickness. So you can see here that skin epithelium is it's thick, where in HIF knockout mice this thickness is decreased, as well hyperproliferation, which we define based on the incorporation of EDU in skin epithelium. So this really proved that HIF-1 alpha is a key factor that drives this skin epithelial pathology, and it's required for tissue for psoriasis development. But we not only observe that HIF-1 alpha is essential to drive skin epithelium pathology, but also HIF-1 alpha control other tissue compartment. And we found that by inhibiting HIF-1 alpha in skin epithelium, we not only block this hyperactivation of skin epithelium, but also we block infiltration of the immune cells, more specifically gamma delta T cells, as well other features typical for psoriasis pathology, like hypervascularization and hyperinnervation. So this really brings us to probe to understand the molecular function of HIF1 alpha in psoriasis. So to understand HIF1 alpha function, we perform RNA sequencing from skin, back skin of either wild-type mice or HIF1 alpha epithelial deficient knockout mice, which were treated with imiquimod. And we found similar phenotype. We found the similar phenotype which we observed in the context of physiological repair, that HIF1 alpha regulate glycolysis metabolism. And we found that by inhibiting HIF1 alpha in skin epithelium, we almost completely blocked expression of the genes that are involved in glycolysis. And we also confirmed this finding looking at the expression of glucose transporter 1, which was significantly increased in HIF1 alpha, decreased in HIF1 alpha epithelial specific knockout mice compared to wild type mice. So this really suggests that there is happening in psoriatic pathology. There is some HIF1 alpha metabolic shift which drive tissue pathology. So to probe the function of the metabolism in psoriasis, the progression downstream of HIF1 alpha signaling, we generated epithelial specific knockout mice where we inhibited glycolysis by knockdown glucose transporter 1. And again, we found similar phenotype what we observed in the context of HIF1 alpha knockout mice, that by blocking glycolysis, we blocked psoriasis development, psoriasis pathology, and keratinocytes epithelial hyperactivation, again by epidermal thickness and hyperproliferation. So this really suggests that the metabolism downstream of HIF1 alpha is essential for psoriasis development, and this was not only important for skin epithelium pathology and hyperactivation, but also for the immune compartment. We found that that glycolysis, glycolytic program not only control hyperactivation of skin epithelium, but also affects type 17 inflammation, where we found that when we block glycolysis, we also blocked infiltration of gamma delta T cells as well. We blocked, we inhibited interleukin-17 secretion by gamma delta T cells. So this really brings us to this model where we found that gamma delta T cells that expressed interleukin-17, which is required for HIF1 alpha activation, which drive glycolysis metabolism. And this loop between the circuits between immune cells and skin epithelium was essential to drive tissue pathology. So we really wonder what's the downstream of HIF1 alpha signaling and metabolism, glycolysis, and which factor is this key factor, is this inflammatory mediator that really drive this immune response and expression and activation of type 17 cells. And honestly, for a very long time, we were struggling to find any specific inflammatory mediators. We look on very canonical factors that drive type 17 response, like CCL20, interleukin-23, interleukin-6, 1 alpha or 1 beta, and we couldn't find any connection. So for why we took a step back and we start thinking that in both cases, either in the physiological repair as well tissue pathology, we see this significant defects in metabolism. So we look closer into glycolysis, which is a very complex process, which involve many different steps. And we really tried to find any connection with psoriasis, and we found this paper which found that lactate as a byproduct and product of glycolysis is highly secreted in the psoriatic patient. So we really start wondering, maybe the lactate is this mediator, inflammatory mediators that close this loop between skin epithelium and immune compartment. So to validate whether a lactate is secrete in our model, we culture ex vivo human mouse skin punch biopsies, either from healthy mice or mice that received imiquimod, so the mice with active psoriatic plaque, and we culture them for 24 hours on the dish in the medium with or without 2DG inhibitor. So this is the glycolysis inhibitor that block hexokinase, which is important in very first step of glycolysis. And we found that indeed glycolysis, we found that lactate is significantly secreted to the medium of mice. Excuse me.
How long are you sitting?
I have a meeting till 2 hours. 2 hours. I'm on the meeting.
Okay.
Sorry about that. So we found that lactate is significantly secreted to the medium in imiquimod-treated samples, and the secretion was decreased after we blocked glycolysis. So this really suggests that lactate is important mediators that drive type 17 response and psoriasis development. So to define the function and contribution of lactate in psoriasis progression, we took two different approaches. First, we treated mice, the back skin of the mice, either with LDH inhibitor, so inhibitor of lactate dehydrogenase, which participate in lactate production, or we also treated mice with inhibitors that inhibit lactate transporter, MCT1, MCT4, together with imiquimod. And we found that by blocking either lactate production or lactate transport, we blocked psoriasis progression, psoriasis pathology, which we observed on keratinocytes hyperactivation, as well infiltration of the immune cells. We see significantly decreased of immune cells infiltration in the skin after blocking lactate production and transport. As well, we found that this was one of the most significant results, that by blocking lactate transport and production, we also inhibit interleukin-17 secretion by gamma delta T cells. So this really proved that lactate is this important metabolic mediators that drive psoriatic pathology. But the bigger question was, okay, we know that lactate participate in psoriasis pathology, but does lactate directly regulate interleukin-17 secretion? So to answer this question, we isolate gamma delta T cells from skin draining lymph node, and we culture them with or without lactate for 48 hours, and we found that indeed lactate was sufficient to induce interleukin-17 secretion by gamma delta T cells. So this really brings us to this model which we presented in the paper, where we proved that the same crosstalk, the same signaling pathway that involved interleukin-17 in HIF-1 alpha is co-opted in tissue pathology, in skin inflammatory diseases. And we also further revealed that inflammation-driven epithelial glycolytic program fuels diseases circuits that both by intrinsic signals that activate skin epithelium and cause this hyperactivation and epithelial dysfunction, but also by extrinsic signals that drive the other tissue compartments pathology, which includes gamma delta T cells infiltration, hypervascularization, as well hyperinnervation. And with that, I would like to thank all the NYU Lab members, especially Shruti for her amazing support, Ipsita Subudi and Alexander Prustypa, who really drive this project, my funding source, my collaborators as well, you for your attention. And I will just use this as an opportunity to mention that next year I'm moving to California to join University of California, Irvine, to establish my independent research group and lab. So if there is anyone on the Zoom who is, or you know someone who is interested in tissue immunity and tissue repair, please reach out. I'm actively looking